Okay, good morning. I would first like to thank the organizers for giving me this opportunity to present our work. Today I'll talk about Oncomer 138, which we think is a therapeutic target for malignant gliomas. Glioblastoma multiforme is the most frequent form of primary brain tumors. It's a highly lethal intracranial malignancy. Most common symptoms include headache, seizure, focal neurological deficit, and change in mental status. If you look at the magnetic resonance imaging of glioblastoma, you can see a central dense necrotic area surrounded by peripheral enhancement, which indicates the highly vascularized area of the tumor. If you look at the current therapeutic options, that mainly include surgery, radiation, and chemotherapy. Despite this multimodality of treatment, the median survival of the patients is only about 12 months. The tumors recur as focal masses. So one of the hypotheses here is that, are these malignancies maintained by the so-called stem cell-like cells or the tumor-initiating stem cell-like cells, which are intrinsically resistant to therapy? So what are these stem cell-like cells? Also referred to as the cancer stem cells, some of the characteristic features of the cancer stem cells include, they exhibit persistent self-renewal, they undergo sustained proliferation, and they have the capacity to undergo or to initiate tumor formation and propagation. In addition to that, the cancer stem cells are very rare within the tumor mass, and they exhibit characteristic markers, which include several of them, and in addition to that, they have the capacity to undergo differentiation to multiple lineages. So we refer to the cancer stem cells isolated from glioblastoma as the glioma stem cells. How do the glioma stem cell look like? In other words, when isolated from the tumor, they grow as tumor spheres which resemble the normal neural stem cell-derived neurospheres, and they grow as spherical bodies. And these tumor spheres exhibit the characteristic markers of the normal neural stem cells, that is the CD133 and also nestin, and these markers are downregulated upon differentiation. But the most important characteristic feature of the glioma stem cells is their ability to undergo self-renewal. That is, every single cell can give rise to a tumor sphere with repeated passaging. And most important of all this, these cells have the capacity to undergo differentiation and give rise to astrocytes, neurons, and oligodendrocytes. So in other words, the glioma stem cells resemble the normal neural stem cells and share several characteristic features of the normal neural stem cells. However, the glioma stem cells are different in the fact that they are tumorigenic. When implanted intracranially, they form massive tumors, and the tumors have the characteristic features of glioblastoma, that is the central necrotic area, and they also show the serpentine pseudopellucid nature. So in an attempt to understand this, since the glioma stem cells are tumorigenic, resistant to radiation, and resistant to chemotherapy, we wanted to compare the expression profile of the glioma stem cells versus the normal neural stem cells, and mainly look at the microRNA expression profile. But what are microRNAs? MicroRNAs are members of small RNA family. They are short non-coding RNAs, 18 to 22 nucleotides long. They control a wide array of biological processes. They bind to complementary mRNA targets and regulate more than 30% of the genes, including transcription factors. And most important, alterations in microRNA expression are known to play a vital role in cancer initiation, progression, and metastasis. So with this as a background, when we compare the expression profile of the normal neural stem cells versus the glioma stem cells, you can see here this is the representation of the data as a scatter plot. Most of the genes fall within the two-fold cutoff range. That indicates that the glioma stem cells are very similar to the normal neural stem cells. However, what you also see is a few genes which are differentially expressed. And we picked one such differentially expressed gene, that is microRNA 138. And microRNA 138 we picked this microRNA mainly because it was the most differentially expressed gene and most consistently differentially expressed gene. And this was upregulated in all the glioma stem cells. As a very first step, we verified this by real-time PCR analysis. And as you can see here, all the 5 patient samples exhibit high level of microRNA 138 compared to the normal neural stem cells. And then we verified this by northern blot analysis. And again, you can see that the expression of the microRNA is downregulated upon differentiation. In order to understand the function of this microRNA, we decided to create a functional knockdown of the microRNA using antigomers. Now, how does this work? Based on the similar way as to how a microRNA, which is fully complementary, binds to the mRNA and leads to the degradation of its target, based on a similar principle, we designed an antigomer. The antigomer is now completely complementary to the microRNA, and the antigomer now binds to the microRNA and forms a short double-stranded structure. And this double-stranded structure is now subjected to RNase action that leads to its degradation. So based on this principle, we designed the antigomer. And what you see here is the top panel indicates the scrambled control and non-specific RNA, which does not bind to any microRNA. And here, when this is the lentivirus expressing the scrambled control. The vector here is lentivirus. So when the cells are transduced with the lentivirus expressing scrambled control, the cells are not affected. They grow and form tumor spheres. On the contrary, when we transduce the cells with the lentivirus expressing antimer 138, you can clearly see that the tumor sphere formation is significantly compromised, and you do not see tumor sphere formation. This indicates that maybe there is a block in proliferation, or maybe it is leading to apoptotic cell death. But before we go into that, we first wanted to confirm that this antimer is highly specific and is targeting only microRNA 138 and not other microRNAs. And as you can see here, expression of MIR 138 is only downregulated, but not other microRNAs. Then we wanted to make sure that this antigomer is not really toxic to other cells. So we transduce the same lentivirus on the more sensitive normal neural stem cells. And again, you can see here with the scrambled control, nor with antimer 138, the neural sphere formation is affected, indicating that antimer 138 is not toxic to the normal neural stem cells, and it is highly cell-specific. So all this indicates that microRNA 138 is a pro-survival oncomer essential for the survival of the glioma stem cells. At that point, we wanted to see if antimer inhibits the tumor sphere formation, what exactly is happening? Is there a block in proliferation, or is it leading to apoptotic cell death? So here you can see, as a very first step to understand whether there's a block in proliferation, we decided to perform a cell cycle analysis. The top panel indicates when you treat with scrambled control, there is increase in S-phase cells, indicating the proliferation is not affected. However, when treated with antimer 138, you can see a significant decrease in S-phase population, indicating that there is a block in proliferation. But in addition to that, what you also see is an increase in sub-G1 population, indicating that maybe the cells are also undergoing apoptotic cell death. At that point, when we perform the Western blot analysis to measure phosphohistone 3, a decrease in phosphohistone 3 confirms that there is a block in proliferation, and the appearance of cleaved pulp indicates that the cells are undergoing apoptotic cell death. Then we wanted to see, we confirmed that the cells are undergoing apoptotic cell death. by caspase activity and exon 5 positive cells. So you see an increase in caspase activity, increase in annexin 5 positive cells, and a final confirmation by increased tunnel staining all indicates that the cells are undergoing apoptotic cell death. In other words, depletion of microRNA 138 leads to apoptotic cell death. So at that point, we wanted to see if the microRNA is functioning as a pro-survival oncomer, how exactly does it work? What are its targets? So in order to understand that, we took the scramble control, the cells transduced with the scramble control or antimer 138, and performed a microarray analysis. And what you see here is the differentially expressed genes, which mainly include c-MYC, BCL2, aurora kinase, caspase 3, MXG1, and BLCAP. And in addition, as a very first step, we wanted to make sure that all this is very reproducible, and we tried verifying the whole thing by protein analysis, so we performed a Western blot analysis. So the microRNA is targeting several pro-apoptotic genes and tumor suppressor genes. I will summarize this by a schematic. So what we think is happening is microRNA 138 acts as a pro-survival oncomer, and it is a master regulator. It directly targets several pro-apoptotic genes, which are essential for apoptosis and blocks apoptosis. And directly targeting inhibitors of proliferation, it allows the expression of genes like cyclin, c-MYC, aurora kinase, which are essential for proliferation and promotes proliferation. In addition to that, the microRNA directly blocks several genes which are inhibitors of tumorigenesis and promotes tumorigenesis. Thus, by blocking apoptosis, promoting tumorigenesis, and promoting proliferation, the master regulator promotes glioma invasion. So with this, we wanted to see what happens in an in vivo scenario if we implant these cells intracranially. So here what you see is we took the same cells, implanted them intracranially, but remember this is an ex vivo experiment where the cells were transduced with the lentivirus expressing the scramble control or the antimer 138 and implanted intracranially. You can see that there is an increase in bioluminescence only with the scramble control leading to the tumor formation, but not with antimer 138, confirming that depletion of MER138 significantly inhibits tumor growth. So far, whatever I have shown is from the glioma stem cells, which we isolated from the patient samples and we cultured in the lab. So one obvious question here is that, what if the expression of the microRNA is a cell culture artifact? And in order to address this issue, we took patient samples, and we obtained 25 patient samples from Medical University of Graz, and we screened them for the expression of microRNA 138, and this was performed by fluorescent in situ hybridization, wherein we developed specific probes to detect mature microRNA. And what you see here is very high levels of microRNA in certain patient samples compared to the others. And later on, when we analyzed these patient samples, we found that the recurrent tumors or the patients with recurrence had high levels of microRNA compared to the first diagnosis. And normal brain does not exhibit any microRNA 138 nor the scramble control. And this was further verified by isolation of RNA. And all this suggests that the pro-survival oncomer is a potential prognostic biomarker. At that point, we wanted to see what happens if whether we can test any of the database So we found one Italian database in the GEO where this database had about 87 patient samples. So we analyzed this database for the expression of 2 microRNAs. So the x-axis indicates microRNA 21. MicroRNA 21 is a proliferation marker and is expressed in high levels in most of the cancer, including glioblastoma. And as you can see here, the microRNA 21 is expressed to the tune of 6 to 9-fold in all the patient samples. On the contrary, if you look at microRNA 138, microRNA 138 classifies the patients into two groups. The red spots indicate the secondary patients, and the blue spots indicate the primary. In other words, the red indicates mainly the patients with the recurrence or the relapse compared to the first diagnosis. So again, confirming that the microRNA is a prognostic biomarker. And then we went ahead and looked at the TCGA database, which had 540 plus patients, but we narrowed down to patients showing recurrence or relapse, and we found that the expression of the microRNA correlates with increased mortality of the patients. So in summary, what we found was microRNA 138 is a pro-survival oncomer. Pro-apoptotic genes and tumor suppressors are direct targets of microRNA 138. MicroRNA 138 is a prognostic biomarker. In recurrent patients, high levels of microRNA 138 correlates with increased mortality. At that point, since it was all glioma stem cells, we wanted to see what happens in a heterogeneous context. So we moved on to some cell lines like the U87MG cell line, which is the most popular cell line. We first started off with multiple cell lines and from ATCC. And you can see here that the expression of the microRNA correlates with increased aggressiveness of the cell line. And U87MG cell line is one of the most aggressive cell line from glioblastoma and most commonly used cell line. And as you can see here, when the cells were transduced with a lentivirus expressing scramble control or with anti-MIR138, you can see with anti-MIR138, there is a significant amount of cell death, suggesting that depletion of microRNA 138 leads to apoptotic death. Then we wanted to see what happens if we inject these lentivirus directly into the tumor mass. So this was the most toughest experiment for us. We started with the tumor size as small as 0.5 millimeters and using the same coordinate, and this was measured by MRI, and using the same coordinates, we tried to inject the lentivirus directly into the tumor mass. And what we see here is by day 26, a massive tumor with this control samples, but with anti-MIR138, you can see significantly smaller tumors, confirming that depletion of microRNA significantly inhibits tumor growth. So at that point, moving away from lentivirus, we wanted to see whether we can develop a chemically modified anti-MIR138. Since the nascent oligo is not very stable, we designed chemically modified antimers with 3 different modifications wherein we modify the backbone. And here you can see, and we refer to the chemically modified antimers as AMOS 138. And similar to the lentivirus, when you treat the cells with, transfect the cells with the control AMOS or AMOS 138, you can see AMOS 138 has a significant effect on the cells. In other words, it leads to apoptotic cell death. And you can clearly see that AMOS 138 effectively target microRNA 138, which is significantly downregulated. At that point, we then decided to transfect these cells. It's an ex vivo experiment. And when the cells were implanted intracranially with the control oligos, you see a massive tumor. But with AMOS 138, you do not see tumor formation, suggesting that AMOS 138 can effectively target 138 and inhibit tumor growth. But injection, we wanted to see whether we can directly inject AMOS intracranially, but that was a physically, meaning it was an impossible task because we could not inject loads of AMOS 138. So we went ahead and made flank tumors, that is a subcutaneous flank tumors, and we injected either AMOS or the control on either side of the flank region. And we can see that with AMOS 138, there is a significantly smaller tumor and much paler tumors. So our current ongoing work includes, we are trying to develop efficient delivery methods. That is, we are trying to develop the convection-enhanced delivery. We are trying to formulate this in nanoparticles and also intranasal delivery. We are trying to use a very novel system of the stealth delivery system, which is a non-toxic delivery system based on concentration-driven diffusion. With this, I would like to acknowledge my group. my group that contributed to this work, and our clinical collaborators and A staff for all the financial support on the project. Thank you.